The Golgi body, also known as the Golgi apparatus or Golgi complex, is a membrane-bound organelle found in eukaryotic cells. It's a critical part of the cell's endomembrane system, and is primarily responsible for processing, packaging, and transporting proteins and lipids to their final destinations. The structure of the Golgi body is characterized by a stack of flattened, membrane-enclosed sacs called cisternae. Key structural components. Cisternae. The fundamental structural units of the Golgi apparatus are the cisternae. These are disc-shaped membrane-bound sacs that are stacked together. A single stack of cisternae is called a dictyosome. The number of cisternae in a stack varies depending on the type of cell and organism, but it typically ranges from 4 to 8. Polarity. The Golgi apparatus has a distinct structural and functional polarity with two main faces. Cis face. This is the receiving face of the Golgi, positioned near the endoplasmic reticulum, ER. Proteins and lipids synthesized in the ER are transported to the cis face via transport vesicles. The cis face is convex in shape. Trans face. This is the shipping or exit face, located farthest from the ER. Processed molecules leave the Golgi from the trans face in vesicles destined for other organelles or the cell's exterior. The trans face is concave. Golgi networks. Two networks of interconnected tubules and vesicles are associated with the main stack of cisternae, cis-Golgi network, CGN, a network of interconnected tubules and vesicles at the cis face, where vesicles from the ER fuse to deliver their contents. It's the entry point for materials into the Golgi, trans-Golgi network, TGN, a complex network of tubules and vesicles at the trans face. It's the sorting station where processed molecules are packaged into different types of vesicles and sent to their final destinations. Vesicles. The Golgi apparatus functions through a continuous process of budding and fusing of small membrane-bound sacs called vesicles. These vesicles transport materials between the ER and the Golgi, and from the Golgi to other parts of the cell. Types. While there are not different types of Golgi bodies in the sense of fundamentally different structures, the organelle is often divided into 4 functionally distinct regions or subcompartments that represent the stages of protein and lipid processing. Cis-Golgi network, CGN, the initial receiving compartment. Golgi stack, the central part of the Golgi, which is further divided into medial Golgi, the middle cisternae where most of the metabolic modifications occur. Trans-Golgi, the cisternae closer to the exit face. Trans-Golgi network, TGN, the final sorting and distribution hub, where vesicles bud off to their destinations. The Golgi apparatus is a key organelle in eukaryotic cells that acts like a cellular post office, modifying, sorting, and packaging proteins and lipids for transport to their final destinations. This process is highly directional and depends on the distinct structures of its two faces, the CIS face and the trans face. Cis face, forming face. The CIS face is the entry point of the Golgi apparatus located near the endoplasmic reticulum, ER. It's the convex, receiving side of the organelle. It receives proteins and lipids in transport vesicles that bud off from the ER. These vesicles fuse with the Golgi membrane at the CIS face, delivering their contents to the first compartment, the cis-Golgi network. This is where the initial processing of the cargo begins. Think of the CIS face as the receiving dock where all the raw packages arrive before being processed and sorted. Trans face, maturing face. The trans face is the exit point of the Golgi apparatus. It's the concave shipping side, located on the opposite side of the CIS face, often oriented toward the cell membrane. After proteins and lipids have been modified and sorted as they move through the Golgi's intermediate compartments, cisternae, they are packaged into new vesicles at the trans face. These new vesicles then bud off and are dispatched to their specific destinations, such as the cell membrane for secretion, lysosomes, or other organelles. The trans face can be likened to the shipping department where final products are boxed up and sent out.